Hi everyone, welcome to the Around the World Innovation in Japan. I'm Masato Yamamoto, University of Minnesota, moderating the session today. Today's webinar will explore the latest breakthrough in the gene and cell therapy research and is a collaboration between American Society of Gene and Cell Therapy and the Japanese Society of Gene and Cell Therapy. All questions will be saved until the end, and then all three speakers will answer questions asked throughout the presentations. You are encouraged to submit your questions through the throughout the presentation using the Q&A tab located under the video player, and I'd appreciate it if you could identify to whom the question is directed to. You may continue submit questions throughout the Q&A discussion at the end of the talks as well. So we have three amazing presenters today. The first speaker is Dr. Hitoshi Osaka. He is a pediatrician and carried out research on molecular pharmacology in the United States at the UCSD. After researching neurodegenerative disease in the Department of Degenerative Neurological Disease in the National Center for Neurology and Psychiatry, Japan, he worked at the Kanagawa Children's Medical Center, and now he is currently working at the Jichi Medical University. Today, he will discuss gene therapy for GLUT1 deficiency syndrome. Go ahead to start the video.
First of all, I will thank the organizing committees for inviting me to this Around the World. My talk is Gene Therapy for GLUT1 Deficiency Syndrome. GLUT1 Deficiency Syndrome is caused by heterozygous SLC2A1 gene mutations, and more than 70 patients are recognized in Japan. Energy starvation causes various symptoms such as seizures, intellectual disability, developmental delay. The ketogenic diet is very tough, and a high-fat diet would cause cardiovascular adverse effects. We established a functional assay at first. Glucose transported into the cell is promptly degraded. Therefore, we incubated 2DG with HEC293 cells. Transported 2DG is phosphorylated and stable in the cell. Then we oxygenized phosphorylated 2DG and measured NADPH level, which is parallel to the amount of 2DG transportation. NADPH levels were well correlated to the disease severity. This system allows us to test the function of GLUT1 and vector evaluation for the therapy. To determine the route of injection, systemic or local cerebral injection, we generated a modified AAV vector with or without a MICTAC. Well-established synapsin-1 promoter was placed at 5' regions for SLC2A1. We used a heterozygous knockout of GLUT1 gene mouse that mimics human GLUT1 DS. For systemic administration, AAV vector was injected into the peritoneum 1 week after birth. For local administration, the vector was injected directly into the bilateral lateral ventricles at 6 weeks after birth. After injection, mice were analyzed for gene expressions, motor function, and glucose level. GLUT1 messenger RNA expression in the forecerebellum. The upper panel shows a vector-specific messenger RNA expression of GLUT1. Vector-specific bands were found in both injection routes. In the lower panel, we compare the total amount of messenger RNA of GLUT1. The best expression was obtained in no-tagged ICV, followed by ICV with tag and intraperitoneal injection. Next, we examined GLUT1 protein expression after AAV injection. A modest level of exogenous GLUT1 detected in the forecerebellum and cerebral Purkinje cells. After intracerebral ventricular injection, a high level of exogenous GLUT1 was detected in the cerebral cortex and hippocampus nearby the injection site. GLUT1 expressions in distant area were minimal. This figure shows the Rotarod test at the age of 10 weeks, corresponding 4 weeks after ICV vector injection. Rotarod test is showing trends for motor function improvement in both injection routes. Especially after intracerebral ventricular injection with no tag, motor function was significantly improved. Next, we determine the core promoter sequences of SLC2A1 and change the synapsin1 promoter to the core GLUT1 promoter. Then we injected model mice with intracerebral ventricular route. Two months after injection, GLUT1 was strongly expressed in the cerebral cortex, hippocampus, and thalamus, but faintly in the cerebellum and brainstem. This slide shows a co-immunostaining of GLUT1 with various cell types markers. Exogenous GLUT1 was expressed mainly in the endothelial cells and followed by neural cells and oligodendrocytes. Rotarod tests were performed at 14 weeks, corresponding to 8 weeks after the ICV injection. The red bar is wild-type mice, the purple bar is AAV GLUT1 treated heterozygous mice, the blue bar is AAV with synapsin promoter treated mice, and green bar is controlled heterozygous mice. ICV with GLUT1 promoter significantly improved motor function. CSF glucose level of vector-treated mice. CSF glucose level was decreased in model mice. After vector treatment, the CSF glucose of model mice was significantly improved by both promoters. Improvement was greater in intrinsic promoter in 10 and 14 weeks. Both high and low concentration improved motor function. We also confirmed that the expression is also maintained in 24 months. Exogenous GLUT1 in endothelial cells was maintained in 24 months. Now you can see the expressions in the vascular wall in this old age mouse in the upper right. As a translation into human, we use the big pig. Its weight is between 20 to 30 kilograms and is similar to the pediatric patients. We inserted the catheter from the needle as a lumbar into the cisterna magna. We can visualize the root of the catheter with X-ray. Magnification can show you the top of the catheter as a radiopaque circle indicated by the yellow arrow. Red arrows are showing the catheter in the subarachnoid space. We will conduct clinical trial in the same procedure. This is a construction vector of this trial, and the MIC tag will be removed in human trial. After 2 weeks of vector injection at Cisterna magna, vector DNA delivery was analyzed in various parts of the pig brain. Broad DNA delivery to CNS was obtained. We detected high level of GLUT1 messenger RNA expression in the prefrontal cerebral cortex, and this expression level was comparable to the level of the mouse experiment, where we obtained motor and biochemical improvement. We detected GLUT1 protein throughout the cerebral cortex, hippocampus, pons, cerebellum, and spinal cord from cervical to lumbar. We also obtained physiological GLUT1 protein expressions at the cellular level. We found strong exogenous GLUT1 expression in the endocellular cells and oligodendrocytes, neurons, and vascular foot region of astrocytes. We are going to conduct a single-center open-label phase 1-2 trial. Vector doses are low group 2 times 10^12 and high dose with 6 times 10^12. Primary endpoint are safeness and comparison of CSF glucose level. These are secondary endpoints: comparison of seizure frequency, seizure duration, and various batteries, and glucose metabolism using FDG PET. This is my summary. We've established the functional assay of GLUT1, and we determined the route of injection, intracisterna magna injection of AV-GLUT1 to be a feasible approach, and we are going to start phase 1-2 a clinical trial 2024 to 2025. These are acknowledgements. Thank you very much.
Our second speaker, Dr. Naoki Hosen, is a professor and chair in the Department of Hematology and Oncology at the Osaka University School of Medicine. Today, he will present his work on identification of cancer-specific targets formed by the post-translational change in proteins and the development of CAR T-cell therapies targeting them.
So, hello everyone. I'm Naoki Hosen in Osaka University. Today I want to talk about the identification of cancer-specific targets formed by post-translational changes in proteins and development CAR T-cell therapies targeting them. So this is my COS slide. So multiple myeloma is a hematological cancer derived from plasma cells. And in spite of improvement of the prognosis by introduction of new drugs such as proteasome inhibitors, immunomodulatory drugs, or antibody drugs, cure of myeloma is still difficult. So the people think that CAR T-cell therapy is one of the promising strategies to cure multiple myeloma. As you know, the BCMA CAR T-cell therapy is highly effective for multiple myeloma. The BCMA is expressed in myeloma cells, but not in other normal cells, which means BCMA is a really nice target for CAR T-cell therapy. But even with the BCMA CAR T-cell therapy, it's very hard to cure the myeloma disease. So the several researchers, including us, are now testing the potential of other targets. So I've been working on identifying multiple myeloma specific cell surface antigens as a target of CAR T cells. But most transcripts or proteins highly specific for myeloma cells are likely to have already been identified because many researchers performed extensive efforts using transcriptome or proteome analysis. But I thought that myeloma-specific antigens formed by post-translational events may have been missed. For example, glycosylation or a conformational change. So I decided to isolate myeloma-specific antibody as many as possible. Then we clarify the character of the antigen recognized by them. So we actually established more than 10,000 clones of anti-myeloma antibody. Then we select the antibodies that do not react with normal PBMC from healthy donors. Because myeloma cell is also a hematopoietic cells, most of the antibodies also react with a normal subsets of normal hematopoietic cells. So, and we got 500 candidates at this point. Then we select the antibodies that react with myeloma cells, but not with CD45 positive normal leukocytes in bone marrow myeloma patients. And we finally found one good antibody, MMG49, is a good candidate. This shows the example of the analysis of myeloma patients' bone marrow cells. So here is myeloma cells, MMG49 antibody react with all of the myeloma cells, but it does not react with CD45 positive normal leukocytes in the same patient's bone marrow cells. Then we identify the antigen recognized by this antibody using expression cloning method. So we generate the cDNA library from the myeloma cells. Then we transduce the cDNA library into the mouse Bacteri cells, which is not reactive to MMG49. Then we see the very tiny cell populations that expressed antigen recognized by MMG49, by MMG49. And these antigen-positive cells were enriched by FACS sorting, and then finally we found these antigen-positive cells expressed integrin beta-7, which means integrin beta MMG49 recognizes integrin beta-7 protein. But this was very strange because pre-existing anti-beta-7 antibody react with many of the normal lymphocytes. But MMG49, which is certainly specific for integrin beta-7 protein, did not react with normal lymphocytes. And also there are several other cell populations that express the integrin beta-7 protein, but the MMG49 did not react with these normal cells, but only with myeloma cells. So in cooperation with the structure biologist, we clarified the mechanism of this specificity. So we first found that, as many of you know, the integrins adopt the inactive conformation like this, or active conformation of like this. And we found that MMG49 react, the epitope of MMG49 is located here, and the epitope is hidden when the integrin adopt inactive conformation. This is why the MMG49 specifically react with the activated conformation of integrin beta-7. So we also found that in myeloma cells, integrin beta-7 constitutively adopt the activated conformation. Normal lymphocytes also express the integrin beta-7 protein, but it adopts inactive conformation in most cases to avoid unwanted cell adhesion. And integrin beta-7 is not expressed in non-hematopoietic tissues. So collectively, MMG49 is highly specific for multiple myeloma. So we generate the CAR T cells using the antigen recognition domain of the MMG14 antibody, and the results on the MMG14 CAR T cells should recognize activated integrin beta 7 specifically expressed on myeloma cells. So when we transplant the myeloma cells into the immunodeficient mice, and then treat the mice with MMG14 CAR T cells or control T cells. As you can see here, in the mice treated with control T cells, myeloma cells grew rapidly. In contrast, when the mice were treated with MMG14 CAR T cells, tumors are gone, showing the strong anti-tumor effect of these CAR T cells. So now we are testing in a clinical trial performed by the Oscar Pharmaceutical Company. And then this study showed that cancer-specific conformation epitopes may yet be identified in many cell surface protein, even if the expression of the protein is not cancer-specific. So recently, we published another example of this type of interesting cancer-specific antigens. The screening strategy is the same. So among the more than 10,000 clones of anti-myeloma antibody, we found one antibody named RH3 react with myeloma cells, but not with normal CD45 positive normal leukocytes in the same patient's bone marrow. And then we found this antibody react with CD90 heavy chain protein. But again, this was very strange because CD90 heavy chain protein was expressed not only in myeloma cells but also the every kinds of normal hematopoietic cells, including hematopoietic stem cells or progenitor cells. But RH3, which is certainly specific for CD90 heavy chain, only binds to myeloma cells, but not with normal hematopoietic cells. So CD90 heavy chain protein is heavily N-glycosylated protein, and we found that when this shows in this experiment, also lysates of myeloma cells or normal leukocytes were electrophoresed and then embodied with anti-CD90 heavy chain polyclonal antibody, which detects all the CD90 heavy chain proteins. As you can see, and here, the electrophoretic mobility of CD90 heavy chain expressed in myeloma cells and normal leukocytes is largely different, but when we remove the N-glycan by treating with enzyme, the electrophoretic mobility becomes the same, which indicate that CD90 heavy chain glycoforms expressed in myeloma cells were distinct from those expressed in normal leukocytes. We also found that in the cells that have immature N-glycan protein, N-glycosylation protein, the expression of CD90 heavy chain protein did not change, but the activity to RH2A3 significantly increased. And so suggesting that the activity of RH2A3 largely affected by the glycosylation of CD90 heavy chain protein. So collectively, these results suggest that the differences in N-glycosylation of CD90 heavy chain may be the cause of RH2A3 not binding to normal blood cells. But CD90 heavy chain is also expressed in non-hematopoietic system. So we checked the binding of RH2A3 in several types of epithelial cells which express the CD90 heavy chain protein. For example, normal skin or normal colon. Actually, these tissues, in these tissues, CD90 heavy chain was expressed. But the RH2A3, the activity was not detected by immunohistochemistry or flow cytometry. And So we generate the CAR T cell using the antigen recognition domain of Rh2 antibody. These cells kills the tumor cells in a specific manner to CD98 heavy chain protein expression. And then we transplant the tumor cells into the immunodeficient mice, then treat the mice with Rh2 derived CAR T cells or control T cells. As you can see here, in the control mice, tumor cells grew rapidly. In contrast, the mice treated with Rh2 derived CAR T cells, tumor growth was significantly suppressed, showing the in vivo anti-tumor effect of Rh2 CAR T cells. So today I show the two examples of the cancer-specific conventional epitopes that were identified that can be identified in non-tumor specific proteins by extensive screening of primary human tumor samples. One is MMG49 CAR T cells recognizing the conformation of protein. And also we also show that Rh2 CAR T cells recognizing the glycome of a protein. So now we are searching for similar targets also in other hematopoietic malignancies and also the various solid tumors. So that's it. Thank you for hearing, and I'm happy to take your question.
Our final speaker is Dr. Shigeki Yagyu. Dr. Yagyu is a pediatric oncologist and a professor at the Shinshu University Innovative Research and Liaison Organization. Dr. Yagyu has a basic and translational research laboratory focusing on developing novel cell and gene therapy approaches for hematologic and solid malignancies. In particular, he is interested in clinical application of cellular immunotherapy, including CAR T cell and the NK cell. Today, he will present the memory-rich CAR T cell engineering by piggyback transposon system for solid malignancies.
Thank you very much for having me here. It's truly honored for me to present our team's work at the joint symposium. Today, I will provide an overview of our research on transposon-based CAR T cells, with particular focus on the piggyback transposon-based CAR T cells. And I'll dive into the manufacturing process and highlight the key characteristics that make them so appealing in cancer treatment. This is my disclosure. This schema shows the differentiation status of CAR T cells. And basically, one crucial aspect is that the differentiation status of CAR T cells determine the functions. Stem cell memory-like phenotype that have the higher proliferation capacity and longevity in the body strongly correlated with the CAR T cell function. On the other hand, the effector-like CAR T cells display rapid cytotoxicity but lose their function quickly due to the exhaustion. It's important to note that the manufacturing process significantly impacts the CAR T cell differentiation. This is a very good example that clearly demonstrated that phenotype and CAR T cells determine the clinical response. In this clinical trial of CD19 CAR T cells for the patient with chronic lymphoblastic leukemia, the researchers divided the patient into two groups, the responders and non-responders, and they tried to find what is the determinant of the clinical responses. There are no big differences in terms of the disease status or tumor biology in these two groups, but there are clear differences in the phenotype of the final product. The CAR T cell product established from the responders showed the memory-rich phenotype with less expression of PD-1. So the phenotype and the presence of early T cell exhaustion in CAR T cell product determines the clinical responses. Therefore, to enhance the CAR T cell functions, the various modifications as listed here have been explored to produce the memory-rich, less exhausted CAR T cells. In these modifications, our teams has been extensively researching the piggyback transpose-mediated CAR T cell manufacturing. Not only its the simple manufacturing system or the cost effectiveness, but also for its favorable characteristics. So over 10 years ago, our team first started applying the piggyback system for genetic modification of T cells. In this study, the GFP and delta CD19 genes were introduced into T cells by piggyback transposon system, and these cells were stimulated and expanded with the anti-CD3, CD28 antibody and IL-2. Piggyback transposon effectively introduced the gene of interest into T cells. However, the transduction efficiency should be improved, and the purification step was needed to obtain a higher population of gene-modified T cells. Moreover, these cells showed a CD62 ligand negative, CD28 negative effector-like differentiated T cells. So this is the first record of the piggyback transposon-mediated HER2 CAR T cells. In this study, the piggyback HER2 CAR transgene was introduced into T cells, and these cells were expanded with ABV-LCL and the cytokines to generate piggyback HER-CTL. Approximately 40 to 50% of CAR T cells showed a CD62 ligand plus CD45 allo plus central memory phenotype. Even though these T cells required the purification step to obtain the higher population of CAR T positive T cells, the piggyback HER2 CAR T cells effectively developed the glioblastoma xenograft model, as you can see in the bottom. As you may know well, the retroviral CAR T cell production involves T cell stimulation step in the initial step. Because of the nature of the viral transduction system, that the retrovirals can only infect activated and proliferated T cells. However, this step leads to T cell differentiation. In this paper, the conventional pre-activation step by anti-CD3, CD28 antibody for manufacturing of retroviral CAR T cells that reduced stem cell memory-like T cell fraction in the final product. Therefore, the excess activation of T cells could lead to T cell differentiation and early T cell exhaustion. Therefore, we have established the strategies to produce piggyback CAR T cells without inducing the excess T cell activation during the manufacturing step. We avoid the pre-activation step using resting T cells to prevent T cell exhaustion caused by intense stimulation with CD3 antibodies. Instead of using hyperactive transposase, which could lead to excess integration into the genome, we chose the original wild-type piggyback transposase to ensure the controlled integration. For optimal transfection efficiency, we carefully optimized the electroporation process and the expansion step using antigen-presenting fetal cells rather than the CD3 antibodies. To support the expansion of electroporated T cells, we supplemented the culture with IL-7 and IL-15. So why do we follow these manufacturing steps? This is a paper about the manufacturing of piggyback CD19 CAR T cells in our group for the first time. In this study, we generated the piggyback transposon-mediated CD19 CAR T cells, and these cells were stimulated by anti-CD3, CD28 antibodies for expansion. However, these cells showed massive cell death by the first stimulation, probably due to the activation-induced cell death, and require the selection of CAR-positive T cells. Therefore, we avoid to use antibodies for the expansion of CAR T cells, and instead, we used the viral peptide pulse PBMC as a feeder cell, just like the concept of the EBV-specific CAR T cell manufacturing. In this study, we made PBMC-derived feeder cells that are pulsed with EBV, CMV, and adenovirus peptide, and the piggyback CD19 CAR T cells were stimulated with these feeder cells. As you can see on the left, we successfully expanded CD19 CAR T cells with excellent efficiency. However, the final product did not show the specificity of these viruses as evidenced by the ELISpot assay, as you can see here in the bottom. And moreover, when we depleted CD19 positive B cells from the feeder cells, these feeder cells failed to expand piggyback CD19 CAR T cells as shown in the right. Furthermore, when we attempted to apply these protocols to generate CAR T cells targeting solid tumors, the results were not as successful. Here, we tried to make piggyback HER2 CAR T cells just by following the previously established protocol for piggyback CD19 CAR T cells, but we could not see any CAR-positive T cells. So, in manufacturing piggyback CD19 CAR T cells with PBMC-derived feeder cells, CD19 molecule on B cells gives a CAR stimulation as signal 1, and co-stimulatory molecule expressed on B cells would mimic the physiological immunologic synapse, leading to a full activation and expansion of CD19 CAR T cells with a physiological tone, not excessive as CD3 antibodies. So we hypothesized that the genetically modified antigen-presenting feeder cells, which expresses modified antigen and ample co-stimulation, might mimic the physiological immunologic synapse that can provide the optimal activation of T cells. To do this, using HER2 CAR T cells as a model, we developed autologous PBMC-derived feeder cells, which overexpressed modified antigen and recombinant T cell activators, and stimulate CAR-transduced T cells with these feeder cells. So we have tried to establish piggyback CAR T cells targeting HER2. So in this case, we divided PBMC, one for CAR T cell manufacturing and the other one for feeder cell manufacturing. Then we overexpressed the truncated HER2 and T cell activators on feeder cells. In this case, we have optimized the electroporation protocol for DNA transposon plasmid into non-activated the resting T cells, and the electroporated T cells were then mixed to the feeder cells on day 1 for further expansion. So we could obtain around 70% of CAR-positive T cells with dominant CD8 positive, stem cell memory-like phenotype defined as CD45LA, CCL7, CD28, and CD95 positive fraction. And notably, as you can see in the bottom, these CAR T cells have scarce PD-1 expression and the low LAG3, but relatively high TIM3 expression, as you can see in the bottom. So obviously, to maintain the memory phenotype of CAR T cells, co-stimulation on feeder cells would be very important. In this setting, we stimulate the CAR T cells by PBMC-derived feeder cells that only expresses modified antigen or together with ample co-stimulation. And as you can see here, the feeder cells expressing modified antigen and co-stimulation greatly induce the T stem cell memory-like phenotype rather than the feeder cells expressing modified antigen alone, suggesting the necessity of CAR T cell stimulation through the immunologic synapse, not only with the antigen CAR complex, but ample co-stimulation. This is the in vivo data of HER2 CAR T cells that control tumor growth. As you can see in the left, the piggyback HER2 CAR T cells showed excellent anti-tumor efficacy in vivo. And when the first tumor cells were inoculated on day 28 to these mice, the tumor was totally rejected in the mice treated HER2 CAR T cells, suggesting the long-term functionality in vivo of the piggyback HER2 CAR T cells. We also wanted to see the direct comparison of the potency of retro or piggyback HER2 CAR T cells. We generated retrovirally transduced HER2 CAR T cells and the piggyback HER2 CAR T cells. Both of these cells have the similar CAR positivity around 20%. And here is the real-time cell analyzer data, and the y-axis shows the tumor growth, as you can see in the magenta line. And when the CAR T cells were added to the tumor cells, the number of live tumor cells were decreased. And notably, the piggyback CAR T cells demonstrated the more prominent and durable tumor control compared to the retro CAR T cells. So we have extensively investigated the piggyback CAR T cells with another target, including the CD19, GD2, EphB4, FLT3, and so on, as you can see here. And we got consistent results with a higher population of stem cell memory-like phenotype with CD45 allele expression. But in this case, we established these CAR T cells from PBMC with just around 30% of CD45 allele plus positivity. Excuse me. So why are our CAR T cells enriched by CD45 allele plus memory-like T cells? To answer this, we divided a PBMC into CD45 allele positive or negative cells, and we made piggyback CD19 CAR T cell from these two fractions. This is the CAR transgene expression profile on resting T cells 24 hours after electroporation. And as you can see on the right, our positive PBMC showed much greater transduction efficiency than our negative PBMC. Then we cultured these cells for 14 days to check the phenotype. Later on, I will mention this as allele plus CAR in the top or allele minus CAR in the bottom. And as you can see on the top, in allele plus CAR T cells, we can see more significant expansion, higher CAR positive T cells with memory fraction, CD8 dominant population, lower PD-1 expression, and the lower expression of CD57, which is a T cell senescence marker, than in allele minus CAR T cells, as you can see in the bottom. So we also checked the immune exhaustion profiles of allele plus or minus CD19 CAR T cells with antigen simulation. And in this time, we co-culture the tumor cells with allele plus or minus CARs at an ET ratio of 1:1 for 3 days and acquired the expression of PD-1 as shown here. Then we added fresh tumor cells for second round and third round and repeated the co-culture and FACS analysis. And as you can see here, the allele plus CAR T cells were less exhausted even by sequential antigen simulation. We also checked the immune exhaustion profile of allele plus or minus CD19 CAR T cells with antigen simulation by RNA sequencing. And as you can see in the right, RA plus curve shows less expression of canonical exhaustion markers as highlighted in blue, and these exhaustion profiles even maintained after the antigen simulation, as you can see on the right. And finally, we performed an in vivo stress test in which the CAR T-cell dosage was lowered to the functional limit so that these CAR T-cells should be maintained and expanded in vivo to achieve the anti-tumor efficacy. And as you can see on the right-hand side, the RA plus CAR T-cell group showed better tumor control. Then we evaluated the expansion of CAR T-cells in vivo by sequential bone marrow aspiration of the mice. So in these two long-lived mice treated with RA plus CAR T-cells were expanded even day 50 after treatment, reflecting the characteristics of memory-like phenotype. So based on these findings, the quality of the starting materials influence on the manufacturing and potency of piggyback CAR T cells, and the selection of the CD45 RA positive PBMC may have a potential to produce more stable manufacturing and favorable efficacy of piggyback CAR T cells. So in conclusion, the piggyback CAR T cells have appealing characteristics as their simple manufacturing process and the favorable memory-like phenotype. And the memory-rich piggyback CAR T cells possesses durable anti-tumor efficacy. The piggyback-based CAR transgene is likely to be introduced into CD45 RA plus PBMC, and these cells were dominantly expanded, maintaining their memory characteristics. So finally, I'd like to thank all the collaborators and happy to have your question and comment. Thank you very much.
So thank you very much for all the presentations. And now we can take Q&A. The viewers, please submit your question on the Q&A chat. I don't see anything right now. So may I kick off the question first with the question to Dr. Osaka. So yes. Then the data basically focus on the CNS system, but I'm not familiar with this disease. So what other organs are affected by GLUT1 deficiency, and how we can treat those diseases by gene therapy?
Yeah, actually, it is a CNS restricted disease, so there is no symptoms outside CNS.
I see. Yeah, that's good. Then that way, you can transduce CNS by the injection directly, and don't have to worry about other organs. That's great. Right. So, any question amongst us? We have two CAR T presenters today. I don't see any question right now. So let me ask another question. Particularly, this is for the the CAR T side, and so Dr. Hosen, how do you transduce T cells currently? Are you using lenti or retro?
Right now I use retrovirus.
Retrovirus.
Yeah, it's just for the convenience.
I see.
For the company. So, but I know that many people in the world is using the lentivirus. So now we are trying to move to the lentivirus.
Yeah. Actually, I think lenti is quite popular around myself. Yeah. And then the question is to Dr. Yagi. In case using the lentivirus, do we have to activate amplification replication of T cells for transduction, or that is the case for retro only?
But do you mean the differences of the amplification?
Whether we have to activate replication
when actually, most of the people using the pre-activation system for the resting T cells before the transduction of the lenti, even lenti or retroviral transduction.
I see. Yeah, good.
Yeah, I can also answer the question. So I was also curious about the pre-activation when we use lentivirus. It's theoretically it's not needed, but
Yes,
many people using the pre-activation. So I don't know the reason why.
probably the pre-activation also induced expansion after the transduction system.
Yeah.
So maybe if we don't use the pre-activation T cells and the lentiviral gene transduction, definitely we can get the expression of CAR T cells, but the expansion was not so sufficiently.
So they may be your feeder cell may be useful.
I don't know. So, but the one option is the combination of the retro or lentiviral gene transduction with our feeder cell system, keeping the memory phenotype.
Yeah.
I have one more question to Dr. Osaka. It means that what kind of AAV vector is suitable for your usage? AAV9 or something? Or currently we see more and more, the tropism modified AAVs. So for the future, those kind of vectors can help your application?
Oh, yes. Actually, the RAV9 modifying some capsid, it is very suitable for delivery to CNS cells. So in this clinical trial, we will use the AAV9 modified vector.
Oh, that's great. That's great. Yeah.
I have a question to Dr. Osaka. So I'm not very specialist in gene therapy, but so is the AAV vector is transducing the every kinds of cells, or it's specifically in a CNS?
Yeah, it actually delivered to the every organs.
Every organ. So
But
is there any expected side effect because some hepatocytes express the GLUT1 so for long years, is there any side effect of overexpressing GLUT1?
Yeah, actually, it is delivered, but we change the promoter, so the expression is not so high in hepatocytes. Yeah, and AAV9 is the easily enter into the CNS. So it means if we inject into the CNS, it's delivered to the peripheral organ. But so we change the promoter.
So you have a very nice promoter that is very specific for the CNS.
Yeah, that's right.
Thank you.
May I ask one more question to Dr. Hosen? You are identifying several CARs, for targets and generating CARs against those targets. But in the meaning of a selection of a single chain motif, is the affinity higher or better, or we have to control?
No, that's a good question. So it's totally depends on the experiment. It's totally different depending on the antigens. So if the antigen density is very high, we don't need so very high affinity SFP. But if the antigen density is very low, maybe we haven't tested, but we may need a high affinity SFP. But it's theoretical. But it's always we tested every kinds of SFP, and it's one of the problem to build up the construct.
Yeah, when in 1990s, Pharmacia started to sell the kit for single chain antibody generation kit. I was one of the users when I was at the Osaka University, actually. Then, they were recommending so-called shuffling to increase affinity. So now, most of the people, particularly for the single chains, just combine two chains together, and that's pretty much it. So I feel a lot of change of dynamics, change of time, but still, some antigens, particularly the low affinity antigens, or low density antigens, probably they may need to optimize the sequence by shuffling or something to target those low density low expressant ones. Yeah.
But I'm not sure what shuffling means, but when you change the sequence, so you overchange the specificity, so that's another problem.
Yeah, I see. Got it. Okay, so yeah, this is recorded, so we may receive some questions later. But anyway, thank you very much, everyone. And here, those who are attending the webinar, thank you very much for attending. And thank you to all our speakers sharing your expertise and for great discussion. And this event is recorded, as I said, and you will receive an email. You all will receive an email with a link to watch the recording again. And it's going to be I don't know how many days later, but it will become available. And we hope we can continue this conversation at the ASGCT's annual meeting during the JSGCT Symposium in the morning of May 8th. And please go ahead to go to ASGCT website and register. And also JSGCT will hold an annual meeting on July 16th to 18th at Yokohama, Japan. So finally, ASGCT values your feedback and welcomes suggestions for future sessions. Please take a moment to complete the survey linked in the chat or scan the QR code on the screen. Thank you very much, everyone.